Okay, so welcome to the second part of this year's Kendrew Lectures. And this second lecture will be given by John Jumper. And John got his bachelor's degree in mathematics and physics from Vanderbilt. And it was followed by an MPhil from Cambridge, where he worked on condensed matter theory. I have to confess, I spent some time doing the same thing and eventually departed in the same way. He then worked for 3 years at D.E. Shaw's Research Institute in New York City, and worked his co-authored papers with D.E. Shaw, where he first started working on molecular dynamics of proteins, and then went on to do his PhD at the University of Chicago and applying machine learning to protein folding and dynamics. Since 2017, he's been a research scientist at DeepMind, where he's now the research lead on the AlphaFold project. And so in this lecture, he's going to tell us about the details of the AlphaFold program and hopefully plans for the future of AlphaFold and related areas. So, John.
Thank you so much. It's been really wonderful to be here and also to hear about all the amazing work that's done here, and really excited about both the work that's enabled by AlphaFold and to hear about the work that very definitely isn't, and really excited about what the future will bring. But let me tell you a little bit about how AlphaFold is made, kind of peel into some of the layers of what makes this work, how it understands structure, some of the things that it is able to do, some of the things that isn't, and maybe try and give some sort of notion in which how does this program understand structure, which parts doesn't it, and really where do we think this will lead in the future? And I always think that AlphaFold is kind of two things. It's a software program that produces accurate protein structures. You put in a sequence, and the structure that comes out at the end is accurate often in many ways. But then it's also a set of ideas and kind of how we build neural networks around proteins. And those ideas we think will impact many more problems and new ideas to come. And that we're really going to look back on this as saying, well, this was kind of structure prediction of single change is kind of the first thing that we were able to do with these tools, but that these ideas and methods will grow and really start to reshape how we investigate biological systems. And so by way of introduction, and certainly with Dimas's great talk, and probably even before, you've heard about kind of the results at CASP and both the progress that we were able to make and the kind of results, and on systems both small and very important like ORF8 from SARS-CoV-2, and very large systems like the RNA polymerase on the left. Well, maybe not very large in the scale of biological assembly, but very large in the scale of protein structure prediction. And I want to really say again that CASP and this notion that we're going to get everyone together and we're going to really see which computational methods work, and we're not going to rely on individual people to hopefully run the same set of experiments or to not accidentally include the test set of proteins in the training set, that we're going to really treat this as a real problem with the cooperation of the experimental community and measure every 2 years how the whole field is doing to understand what works and doesn't, has really been an essential and important part, both in terms of the historical development and understanding where it is. And I would also encourage you all to read the papers that have come out from the CASP assessors, who talk in detail about the results and what they may mean. And so similarly, we have not only put out, after we built this system, open source code, but we've taken this attempt at folding all the structures of humans and 20 other model organisms. We have plans to expand this to approximately UniRef 90 to cover fold space, and we think that this will drive us into this really interesting regime in which structure becomes abundant. And a really interesting question and kind of research is, how will research change? As it used to be that the line between genomics and structure is that UniProt grew 3,000 times faster than the PDB in terms of number of depositions. And so we're at this, the genomics revolution had given us really sequence abundance. And AlphaFold is a tool to turn that into structural abundance. And so now I think we'll see this interesting regime where we start to see what new science can be done in the era of structural abundance in terms of homology, in terms of understanding species, in terms of understanding human biology. And we think that really this is kind of an exciting and transformational thing that will change our relationship to structure and biology. But to dig into how it works, and I think the kind of core point I want to make at the beginning is that we have known a lot about protein structure for a long time, thanks to some incredible biophysics work by a large number of biophysicists, a lot about evolutionary biology. We've really understood, quite a lot of facts about proteins. But what we've lacked is an ability to synthesize those into the right structure. That it wasn't so obvious. It's not a simple equation that finds, say, the minimum free energy structure. And when we started this work, and when we started with AlphaFold 1, we were doing what was a common approach in the field where we would have a component that was built out of neural networks. But that was really an off-the-shelf system from computer vision. And then, it was exactly the same type of thing that you might do for image recognition. And then around it, we built all our protein knowledge. And we put the amino acids here and kind of optimization of the chain there. And we wanted to build the protein structural knowledge around the deep learning system. And with AlphaFold 2, what we had started to do almost immediately was to say, no, this belongs inside the network, that this is the main component. We need this to understand proteins. Now, how do we take what we understand about proteins and build it into the core components of the network? How do we build pieces that want to learn about proteins so that when we show it the incredibly valuable data from the PDB, which is inherently limited, It's not normal to do machine learning where every data point represents a PhD's worth of effort, This is very, very, very different from image recognition. How do we think about how we build these insights into the structure of our network? And we talk about some specific ones, and I'll talk about some specific ones in the talk, but we needed to build a kind of neural network technology of proteins, and that turned out to be extremely, extremely efficient. And so this goes in kind of a long line of research in machine learning and in neural networks that uses this jargony term inductive bias, that there are certain problems that are easier depending on how you connect your neural network, how you build the individual pieces. And you see this very, very prominently in the enormous success of what are called convolutional networks. And these are like convolutions in image processing, operate on a very small patch of an image and build that up. The analogy in proteins is things that operate on a very local patch of sequence. And so this was how AlphaFold 1 was built, was around these convolutional ideas. And then you build more and more, and you hope that it learns higher level structure. And that reflects well the primary sequence, the notion of kind of local linear motifs. But it represents very, very poorly the 3D spatial structure. That at least at the end of this process, things that are 200 amino acids apart may be right next to each other, must be right next to each other. And so if you think about things that are oriented around, say, very, very local things like convolutions or recurrent networks, then you're not really capturing that aspect of spatial structure or geometry. There's been also a lot of great work on graph networks, things that embed in this notion of connectivity. But the problem is you must start with the answer. If you knew everything that was close to each other, you wouldn't need AlphaFold. And so we base it around these ideas in attention networks. So this is a technology that's become very, very powerful recently, where you, in essence, allow the neural network to build its own connections of different strengths between different parts of the chain. And what we observe in AlphaFold is that as it learns about structure, it will start to build the connections of the pieces of structure it knows. And so this gives it really natural connections between the pieces of structure it built. And we originally we said, okay, we'll put in attention and this will be the right language. And the kind of off-the-shelf attention networks didn't really work at all for this. We didn't get an understanding. We were missing quite a bit more. We had one right notion, that there should be some notion of spontaneous connection that develops within the network. but we were missing lots of other notions of geometry or how does evolution fit into this prediction and everything else. And so we really needed to go back also to this kind of picture, and I liked this picture that has emerged, especially in the picture is very much older than the structure prediction community, but that it is important for structure prediction. And if you think about the causality of protein structure and evolution and everything else, there's a very causal direction. The sequence causes the structure, right? That's what Anfinsen said, and via physics. And then we all know that the function causes the patterns of evolution by preserving function, and that structure changes slowly over evolutionary time. And so the causal direction is kind of that the structure causes the evolutionary history, or at least the patterns observed across evolutionary history. And so evolutionary structure prediction is to kind of be Bayesian, to run the causality in the wrong way, and to take this pile of evolution, or to take the natural sequences that you can find via evolution and say, well, what protein structure must they explain? And here this turns out to be very powerful and not kind of in the way that had been done before, in the very literalist will build the evolutionary correlations and then we'll correct for the direct versus indirect coupling effects, but rather a more general method where we present this as another useful piece of information, that the multi-sequence alignments that we can collect should tell us quite a lot about the structure by reversing the causality arrow. And so when we start from the input sequence for the protein, we use very standard tools, jackhammer and HH-Blitz, to look up a multi-sequence alignment, the evolutionary history, and just really present that as a raw piece of information for our neural network. And so throughout AlphaFold, and there was a very interesting question when talking to students about, well, what are the roles of templates, for example, which we also look up. I'll talk about, and we pass in, we make templates, for example, we pull also from structured databases and we present them just as input. And that goes essentially, that's presented as if it may be interesting to the network rather than as something you thread over. But maybe the really important part is in the middle. And I'll see if I can't use that. But in the middle where you see the residue pairs. And here, this represents almost nothing at the beginning. We put no information at the start of the network. It's more or less the sequence distance of each pair and some other very minor information. It knows very little about the structure unless, except when the templates tell it. But at the beginning, this is encoding a notion of kind of physics or aligning with physics, that we know that physics is largely pairwise, that we know that interactions are very important spatially. And so we build this structure in of pairwise terms that we hope that the network will ultimately use in order to think about residue pairs. And then as we start to build our architecture and we really kind of put the templates into the pairs, and we're missing an arrow there. But as we think about this, we start with very raw information. Here are the sequences and the multi-sequence alignment. Here are all the pairs of residues. And then we're going to put this through a block that we call EvoFormer that we had built specially for this task, that starts to process this data and makes newer, richer, more refined forms. And I really haven't told you how it processes much. And I'll tell you only a bit, because the important point is really, because of this principle of end-to-end training, is what happens at the end. So you see, after we process it through something I haven't told you much about, we produce these very refined representations of the multi-sequence alignment and of the pairs of residues, and then we put them into what we call the structure module. And this is a module that we made to really encode the fact that we are doing 3D geometry. And so it has explicit notions that every entity is a residue with a position and an orientation. And this operates in 3D space and builds that final structure that you see. And of course, at the start of training, it builds a very terrible structure. It will look nothing like a protein. In fact, it won't even have a connected primary sequence. But we can now compare this to an experimental structure in the PDB and find a direction to improve it just a bit. And so what we do is we back propagate. So we start from an error in that structure, and then we correct each part of this to give a slightly better answer and do that iteratively. And so what gives meaning to this pair representation? So it starts with a bit of meaning. It looks like residue pairs, but it gains more meaning at the end because we use it in a very specific way. At the very end, we'll ask each individual pixel at the ij location, what is the distance between residue I and residue j? And we will produce that as an output. So in the course of this neural network, we basically build the blocks around what we want to communicate, how we want information to exchange. And then at the end of the neural network, we give them meaning by asking them a very specific question. And because we've trained this end to end, you can ask really complex questions like, what is the structure? And then you can find all the different places back in the network where you can tweak a little bit to do better. And if you do this a million times in a row, you actually end up with a very, very strong system. And then we add to it an extra notion where we're really trying to push it around refinement. So one kind of notion that is a little bit missing in here is that, you want, when you're predicting a structure, it's often the case, you'll know certain things. You'll be able to build pieces of the structure, and then you need to build more newer pieces. And so what we will do is we'll take the answer from this network and what we call recycle it. We will push that back through the network a second and a third and a fourth time, which tends to produce a bit better answer. And each time it's trying to improve its answer. And so this is a bit also like self-templating the network. One useful template that you may want to use is your answer from a previous round. And out of that, we also pull additional outputs. Of course, the confidence, the PLDDT that Dimas talked about, as well as this pairwise confidence of predicted align error. And this will tell you, this is something you can look up online how to do it, but will tell you when you should trust the domain packing structure, especially of this network. But to kind of dig into the ideas, and I think I really want to dig into one of the core ideas of Evoform, or even some of the core. And it's really important before we go into that to note kind of what parts of what we did mattered. And so if you look at this table, and this is the GDT, that's a measure of structure accuracy difference. Each line of each row of this table represents a separately trained AlphaFold model, trained from scratch from the paper, for the paper. Almost exactly the same as the AlphaFold that we used in CASP, except self-distillation training, which was more expensive. We treated it as an addition rather than a subtraction. But each one of these says, if you remove some idea and train a new network, how well do you do? And you can see almost all the ideas, removing them is bad, right? Across 8 or 10 ideas. In fact, one of the closest to not being bad is to remove templates. The model is really very insensitive to templates in most cases. But what's important to say is that all of these pieces represent different ways in which we tried to align how we built this neural network and the individual pieces of the neural network with our understanding of protein structure. And all of it mattered to some degree, and it starts to matter non-linearly. So each of these are much, much smaller differences than, say, the difference between our CASP AlphaFold model and any other model that was entered into the competition. That would be more like 20 or 30 on this baseline. But what we see is that all these ideas added some, and in combination was extremely powerful. But the important part was really kind of the methodology of doing this alignment. The details matter. And it really sat at the intersection of understanding of protein kind of physics and geometry and biology together with understanding ML and networks. And that's one of the reasons that DeepMind was such a great place to do this work, is that this is a really wonderful computational problem that sits squarely at the interface between these techniques. And so to give an example, kind of a little vignette of some of the ideas that we put in. So this is one that's quite beautiful called triangular attention. So if you remember, that matrix is supposed to represent all pairs of residues. And so we have methods in which we are exchanging information between them or between two elements. And you can think of an element as representing an IJ pair. So if you exchange information across a row, you'll exchange an IJ pair and maybe an IK pair. And each of those you can think of as two legs of the triangle, or if you want to think about the distance between them as what this network represents, then there's a missing piece, right? That the relationship between I and J and I and K is very strongly determined also by the relationship between J and K. This is, say, a geometric point of view. The triangle inequality says that, I guess it labeled here as A, B, and C, but if A and B are close and A and C are close, then B and C must also be close. And so rather than try and put this in a very handcrafted way to say this must exactly represent a distance and we shall push the triangle inequality into it, we built in triangular communication And we built a new piece that I won't go into the details, where the network updates itself with many, many small triangles of residues. And we find that by adding this kind of communication pattern rather than a hard-coded rule, we do observe much stronger obeyance of the triangle inequality by the final output of the network. And we notice a small increase in accuracy. And so, and I should say, by the way, the pairwise distance prediction obey the triangle inequality. Obviously, all coordinates must because they're in space. But we can start to build in kind of these notions we know, but in interesting and exciting ways, and figure out how to build that into the structure of the network and the learning system. So that was kind of a very physical and proper thing. We thought about a good physical geometric fact. We put it into our network and we felt good about it. The structure module is an example of the opposite. We're going to do something horrible and then we'll feel bad about it. So we're going to start from the sequence of the chain and looking at individual residues represented here by triangles, position and orientation. We know that there's a very, very strong restraint, that you can think of this in terms of the Ramachandran angles of the backbone. But this is a very, very bad representation to work with in neural networks in a lot of ways, because thinking about all those individual twists, like if you have, for example, a split domain that goes off for hundreds of residues and comes back, then if you think about it in the kind of natural torsional space, the natural Ramachandran space, then you have kind of a 200 or a couple hundred residue excursion to get back to a couple angstrom interaction. And that's quite terrible. So we just kind of ignore that. And so we treat each individual residue, we call it a residue gas. And we treat each one as a freely optimized thing. And you can, if you look carefully over the iterations in which we optimize this within the structure module, you can see it kind of has an unphysical breathing, and the helices look a little bit too big at the beginning and some other things. And what you're really seeing here is that we're ignoring a good, proper biophysics fact in order to make it easy for the network to optimize all pieces simultaneously. And so over the course of training, it observes that I and I+1 residues are always in this geometry consistent with the peptide bond. But we treat that as a learned fact to allow the network to optimize. And so in this sense, we're kind of trading proper biophysics for computational expediency. And so making these kinds of decisions are what enable ultimately this network, which does a very surprising thing. It has in total 192 blocks of computation. And for a protein of almost any size, at least out to the couple, maybe 3,000 residues that we've tested it with, it proceeds in a finite number of steps to build this structure, right? This is very unreasonable from kind of a physical folding point of view, where protein folding times depend on transition state energies and are highly variable. Here we're saying you have 192 blocks, and so we have to put in these kind of things that favor the computer science aspects of it, the parallelism, over maybe the physical aspects. And so working through all of these, and if you pull these types of constraints correctly, this is part of what builds a system that understands proteins and enables the learning to occur. So in another way to think about this is that DeepMind is about building very general learning systems. And there's quite a lot we don't understand about proteins. And we're kind of say, well, the network will have to understand it. And there's some things that we understand well that we can kind of build into, especially not exactly hard-coded rules, but how we exchange information to enable this type of learning to occur. Now, we also have observed something really, really interesting in terms of thinking about the PDB. It's a wild place. It contains the combined output of the entire structural biology community, which includes everything from exciting ligands to post-translational modifications to all sorts of weird and wild things. And the neural network is trained on all of them and is presented only with the primary sequence of one of the chains. And so it has to be, because this is such an underspecified problem, the neural network has to be tolerant to underspecified conditions, right? It can't fail on every protein that has an obligate heme just because we didn't tell it there's a heme. And so we do observe actually quite strongly something that looks like at least tolerance and maybe even kind of knowledge of pieces of structure that are not specified. So on the left, you see a zinc binding site where we don't predict the zinc. This is overlaid from the crystal, but the side chain geometries are perfect for the zinc. We also see very commonly on homomeric proteins that the model will in fact, even though AlphaFold was only predicting one of these chains, and we just overlaid on the trimeric structure, it produced a highly intertwined trimer in a configuration that makes no sense for a single chain because it had to do that. It saw a tremendous number of homomeric proteins in the PDB, so it had to have some ability to reason over this missing context. We can go further in trying to understand what the network kind of thinks about structure in so much as it thinks. And the way we do this, or the way we tried to kind of peel open the black box, is to take a trained AlphaFold network, which is called frozen, right? We're not going to change it. But then for every layer of the network, we are going to ask it to produce its answer right now. And we train 48 individual predictors that every point in the network go and predict the structure as of that moment. And so you can think of this as a way to manifest AlphaFold's belief about structure at that moment. And when you do that, you see some really interesting patterns. And this is OR8, which is one of the most challenging, one of the more challenging proteins that AlphaFold has predicted the structure of successfully. And what you see over the individual layers, obviously it figures out the secondary structure quite quickly, but you see something that looks almost pseudo-dynamical, right? Quite reminded me, other than the very early layers, of what I had seen in molecular dynamics, is it starts to fit and pull pieces together. And as it obtains the correct geometry, it tends to lock it into place. But we see a lot of evidence that AlphaFold is kind of exploring with a very concrete structural hypothesis, and then building and maintaining the parts of that hypothesis that are true, that are reinforced probably by all the interactions. And if we look at this, and you can see a very unphysical structure at the beginning, obviously, but if you look at this on a very large 2,000 residue chain, you can see that some domains are built very early, and then some are built very late and presumably refined against the kind of information that has been discovered in intermediate layers. And when we go and we look at predictions of distance, for example, we'll see some pieces of structure emerge confidently, and then we'll see the kind of neural network information exchange will happen exactly over residues that it knows are close in space. Whereas when it doesn't know where a residue is, you'll see much more diffuse information transfer. And so we have in this system, and something that we didn't quite really build in, right, certainly not over the individual layers, is a kind of iterative learning, that it is kind of searching, kind of building at each stage on its piece of knowledge and doing it in a way that's reliable. And what would really go wrong if you try and do this by hand is that you would make wrong decisions and be unable to correct them. And the network seems relatively robust to that over the course of its evolution. It's something we're still digging quite a bit more into. And you can see these are the representation. The y-axis is the structure accuracy, and the x-axis is this is actually T1024 is a slightly different protein that was very easy. And you can see for very easy proteins, very early in the network, AlphaFold gives a correct structure and sticks with it. Whereas for a very difficult protein like OR8, which is labeled T1064, because that was what it was in CASP, you can see that it requires many, many layers in order to come up with a structure. And in fact, probably we stopped a bit early at 192. And so this is the mechanism that we have for what we call recycling, is basically a way to give the network more time to think about very difficult proteins. And so and we're starting to see kind of this iterative process that has emerged spontaneously within AlphaFold, and it's starting to make us think about how we should build the next generation of systems. Now, the impact of AlphaFold, I mean, I feel a little bit bad about trying to tell structural biologists about this, because really I'm learning from structural biologists about this. But one of the things that we want to talk about, and as we decided to fold the human proteome, it was a very exciting thing, and we were very unsure about exactly how much we would add to the structural coverage, right? The human proteome is extremely well studied. But we do see, as Dim has said, about a doubling if you compare the experimentally known structures to now the high confidence structures. Here, the gray band represents anything with a strong template. And so this represents even more what you might expect to do very well on by template modeling. And you can still see quite large increases in coverage in both the very high and high confidence bands. So even across an organism like human, we're seeing a large increase in structural coverage. And I would point out especially the integral component of membrane proteins. AlphaFold is in general as accurate on membrane proteins as it is on globular proteins. Often the evolutionary signal is very, very strong on membrane proteins. So we add a particularly large amount of coverage to that class, as would be expected. And I'll talk about a little bit later why these bars don't go all the way to the right, why the high confidence residues or even medium and low confidence don't cover the entire chain, and the rest of it is very low confidence. But it's also worth saying that AlphaFold has had this really incredible impact in terms of enhancing experimental structure determination that we've been very excited about. And on the right is a HAMP domain solved by Andre Lupus's group, and this was actually in CASP, but at the time they entered it in CASP, they didn't know the structure, and they had 10-year-old X-ray data sitting on their shelf. And so it was the CASP organizers thought it would be an appropriate challenge to really, it's so blind, the experimentalists don't know the structure. And AlphaFold was able to produce a quite good model you can see in green and phase these data. We're also seeing this having an impact in cryo-EM, especially the low resolution cryo-EM. This is the AR9 from the AR9 complex of Peter Lehman's group. And here what we're really seeing is exciting things where AlphaFold is kind of a very nice complement to cryo-EM because there are a lot of medium or low resolution regions that AlphaFold can give really, really high, can really give atomistic detail to this, in addition to some really exciting work, and I don't have it in the deck, from people like Tristan Kroll, who's had this amazing thing of using AlphaFold for proteins of unknown sequence, but he determined the sequence from a cryo density and some other things. Really exciting work. And we're even starting to see this in NMR, and Guy Montaglioni and others that have worked on how AlphaFold structures can be used for NMR peak assignment and to improve NMR coordinate determination. And so it's really exciting to us, both for kind of the immediacy of the feedback of, we've definitely got it correct. There's little doubt on either of these. Certainly there's no doubt in molecular replacement, but also the ability that these kind of work synergistically, that we're using this to learn about more and more complex biological systems, and certainly will become increasingly important in the era of these amazing cryo-EM complexes. But the other part of the story, the missing part, right? Why does AlphaFold not give confident structures for everything? And I will say one of the scariest parts for us is when we first started folding or predicting the structure of random proteins from UniProt and random human proteins, is they looked a lot worse than the things that we predicted from PDB, right? And this, I don't think would have been too much news to experimentalists, but it was terrifying to us on how many large, terribly ugly segments we would have, kind of like the ones at the bottom right for that protein. And this made us worry very much that maybe AlphaFold was possibly the results were more special to the kinds of things that are already easy to structurally determine. But then we started to really notice that when we looked at the regions that had very low confidence, they corresponded quite well to UniProt disorder annotations. And in fact, if you look at the bottom left, and you can see on resolved residues in PDB, a very high percentage or high confidence. And, a lot of PDB has strong templates, but still very, very few say predicted confidence less than 50, which is a quite low number. Whereas if you look at the unresolved regions of PDB structures, there's a very high percentage of PLDDT less than 50. AlphaFold was very unconfident. And even some of the places which are high confident, some of those may be mistakes, but some of those may also be regions that one resolves, say, due to domain motion. And when we look at the human protein, we see something in between. And so we started to realize and think and want to measure to what extent is AlphaFold saying, I'm not sure, I'm very unsure, really. Well, how much does that mean that there's simply not a structure defined? And really, in many cases, it will turn out to be true. And we believe, for example, that you can identify the domains in the protein in the bottom right, but the ones in between we don't expect to be deposited in PDB, certainly in a nice globular structure. Now, a certain kind of caveat to that note is that disorder means many things, and we use in the paper unstructured in isolation. You can see example in the top right where AlphaFold correctly predicts the domain of this chain. Blue would be the AlphaFold prediction, green would be the experimental structure. But we have very low confidence on the coil region that goes across it. But of course, this is resolved in PDB because it's wrapped around another domain. And so this is something we would call unstructured in isolation, but that doesn't mean it will never appear in PDB in any form. It's just not going to be kind of a globular or well-formed structure. And we're really interested in understanding those. And I'll talk about some work at the end. And we can go ahead and quantitatively measure that. Left was from the paper where we looked at precision and recall of certain thresholds on how much you should believe that this indicates your protein is disordered. And we saw quite good numbers, but we're obviously not experts in disorder prediction. And after the AlphaFold paper came out, some really great work by Blint Mazaros and Norman Davey showed that really actually AlphaFold is a very strong disorder predictor. So it's a slightly complex graph on the right, but if you look at the purple line, that would be a very commonly used disorder predictor, IOPRED2. AlphaFold, various AlphaFold derived measures that they did are the other curves, and you can see a much more favorable area under the curve for precision recall. Now we want to be cautious here that IOPRED is a very fast disorder predictor, but is not the most accurate. And this needs more benchmarking, and work is ongoing. But we think it's quite promising, and we think it's a really good sign that at least on well-studied organism classes, that it's very likely that AlphaFold lack of confidence is a very good sign for disorder. Now, the other kind of point that was very obvious is that single chain structure prediction has quite a lot of limits. And so we really wanted to move and go toward AlphaFold for complexes. And this would work we'd actually done starting before even the paper came out. But it was interestingly, as soon as the paper came out, there was some work, there's the proper way in which you retrain AlphaFold and you show it protein complexes and you expect this to do well. But it turns out that the single chain AlphaFold actually is surprisingly good at predicting, especially homomeric, but also heteromeric interfaces. And this was kind of announced to the scientific community in a series of tweets from Minkyung Baek and from Yoshitaka. And they basically said that there's two different methods. Adding a gap or even adding just a disordered linker sequence is enough to make AlphaFold sometimes produce very confident complexes predictions. And some analysis out of Arne Olufsen group claims that probably this is the currently available state of the art in complex prediction is simply to try and trick AlphaFold into thinking it's one long chain, right? And this is a really kind of interesting and emergent property. And I think it points to kind of AlphaFold isn't aligned in the right way, right? If you think of AlphaFold as a memorization system, this is very confusing because it's never seen a complex in its life. Now, it has probably, I guess you could say, it's seen proteins that perhaps got fused at some point in evolutionary history or split in evolutionary history. But still, you would be relatively surprised that it can do these things. But it is aligned with kind of the physics of interfaces, the geometry, everything else. And we think even for this, it's not even using any type of co-evolutionary signal based on the way the inputs are constructed. And yet, because of the way we built this neural network, it has this understanding that also is the same type of physics that drives individual protein domains and multi-domain interactions are the same type of physics that drive complexes. And so this was really interesting. But we also had work coming out, and this was work that came out, I guess, last week, where we released a system where we had adapted AlphaFold. So we changed the inputs to have multi-domain complexes, the training in terms of how we present now presenting and how we emphasize the complex interfaces, the loss, basically neural network and training details, and trained essentially the same AlphaFold architecture from scratch on complexes and obtained, you can see on the left, a much stronger system. This is DocQ, which is a metric of docking quality, much stronger than linker, gap-based, these types of hacks, regular AlphaFold systems, and starting to produce, and these are cherry-picked examples, but some really compelling examples of complexes. In fact, even talking before and with the linker, I think the gap version, people have seen really compelling complexes, but we think this is quite a bit more accurate. And diving into the accuracy, we find actually only a bit more accurate on homomeric proteins, but on our test set, quite a bit more accurate on heteromeric interactions and with this interface PTM, which is a good confidence measure. Now, this is still, I would say, this has more limitations, not just known stoichiometry. Sadly, it performs poorly on antibody interactions, we believe. We need to assess that more. We don't yet know why. But also, I'd say one of the biggest differences in kind of use from AlphaFold is that if AlphaFold says it doesn't know the structure, that's probably because there isn't one. That's not at all true for this system. So it will still miss quite a few interactions we expect. And so this is an area which we think things will improve rapidly. So what's next? And there are many things next, but I wanted to point out kind of three really big classes of challenges. One is to extend AlphaFold to the other major areas of structure and structure prediction. We want to increase multimeric accuracy, and there's already great work out of the Baker Lab on interactions, but to really start thinking about getting to a complete interaction map. We want to think about nucleic acids and ligand interactions, as well as possibly RNA structure, and of course, conformational diversity. I mean, one of the big limitations, we have no control over whether AlphaFold makes an apo or a holo structure. In many cases, there should be enough data in the PDB to at least pick out a lot of functional states. Roland Dunbrack did a great analysis of what we produce on kinases, and we produce a nice mix of DFG in and DFG out, but we should be producing both of those for all the kinases, right? And so these kinds of things, we're not yet there, but we think that this is something that can be extended in the same sort of ideas, and maybe one or two more will be needed. What we're calling the structural metaproteome, that when we have 100 million structures, what are we going to do with it? And what are we going to do in the era of structural abundance? And one of the answers will be that bioinformatics will surely, at least for things that are encoding regions, will become part of structure. You'll always have the choice to use structure in bioinformatics. But we think there'll be new tools and ways of thinking, and how do we think about having 100 million structures with high reliability? And we think that this will lead to new areas of investigation in science and everything else. And then finally, AlphaFold is not sensitive to point mutations. It finds the functionally encoded structure. And so, questions about how do we think about changes in structure and stability upon point mutation? How do we think about binding affinity and its changes? How do we think about post-translational modification and its effect on the structure? All of these are really important areas for future work. And we think really starting to push outward this beachhead we've got into computational tools that give really powerful predictions on these systems. And so with that, I want to make acknowledgments, and I want to say, both the teams that Dim was pointed to that worked on the methods and the human proteome, and especially Imble and its partnership, and is responsible for kind of a lot of the great things in how we present this data and how we think about displaying disordered regions, that I think is really shifting some of the conversation even in how people think about their proteins in terms of these disordered regions, that they're right up in your face. And they've been really great partners in making this work available to the community. And with that, I'll take questions.
Thank you, Joe. Great talk. Questions? Wait for the mic.
Yeah, thanks. Great talk. I guess my question is a pretty obvious one, but how do you plan to overcome the limitations of basing your network on evolutionary and sequence data? That is, when will AlphaFold become a universal protein folding predictor? Do you envision it predicting proteins that have unnatural amino acids, for example?
I think it's a really good question. I think there's a couple different ways to think about it, and certainly my own thinking of this evolved over the project, because I came out of the biophysics community and I said proteins fold due to physics. We should do that just right. Obviously, we didn't exactly in that way. But I think there's 2 points. One is, for kind of most natural proteins, genomic coverage is not an obstacle. Like for basically no proteins that appeared in CASP of the 100 proteins. And you can find cases, and I think when you get to understudied organisms or things that come out of metagenomics, maybe this will be a larger issue. But for a lot of natural proteins, it's not an issue. I think for getting kind of this overall 1 angstrom fold, in most cases, that's not an issue. Where it really becomes a deep and very important issue is when you get into multiple states and then you want to assess really phase transitions or the energetic, the delta Gs and delta Gs between them, right? And I think when my kind of biophysicist picture of mutations is that they don't change the structure, but they do change the overall free energy of that particular basin. But that overall free energy is dependent on lots of other factors that AlphaFold doesn't know about. It's, pH dependent, it's salt dependent, it's all the other things dependent. And so when you want to get to a system that's really kind of qualitatively telling you that it's shifting between these two, you have to say under what conditions. Maybe you can say before that, okay, well, this is causing this delta G of this state. And then you also have to talk about, well, okay, well, this state exists and that state exists. These are the two that we're comparing. And I think post-translational modifications will be largely the same way. Now, you can think of it as some ways an induced conformational change, but you're almost kind of judging states that exist. Now, some of them exist at a positive free energy and you don't see them, but that's kind of how I think. So I think you will have to get into, as Dimas talks about, this more physical energetic picture in order to answer the biological questions you really had. Yes, the structure is different as deposited in PDB, but that's really due to a quite small shift in a very finely balanced delta G, in my view.
Great talk, John. I was wondering, is there any way the tools you've built, could they be used the other way around? If I want to design a protein of a given structure, perhaps even function, could you provide me with a sequence?
So there's been quite a lot of work external to DeepMind related to this. So yes, or at least it can contribute to. It doesn't directly and immediately solve it. But what you can see, so what we do know, first of all, if you take any design sequence that appears in PDB, AlphaFold normally gives a very, very good structure prediction for it with no multi-sequence alignment, no templates, because the sequence is very, very clear toward the structure. There's been other work in can we optimize and can we make a sequence that AlphaFold thinks has this structure, and then will it actually have this structure? I think even some work with computational validation went on archive about that, maybe today, maybe yesterday. But and there's also been work on kind of back propagating through a neural network the kind of hallucination results. I think that certainly AlphaFold will do a better job, or very likely AlphaFold will be an improvement in those kind of systems that use a structure predictor in the middle. I don't expect that alone will solve all problems immediately, right? So AlphaFold isn't normally, in the same ways AlphaFold is not normally a stability predictor, right? It doesn't pick up point mutational effects. That's an obstacle to these types of things, although not a complete obstacle. And so I think it will be intermediate, but I think it will enable kind of I'll also say the kind of learning systems that you put into that, probably you should put in the architecture that AlphaFold uses, right? So the kind of neural network ideas will carry over quite straightforwardly. Using AlphaFold as a subcomponent of the system, and kind of to Dimas's point is moving away from this end-to-end learning and just saying, we'll just put a component that we built beforehand. And so that's somewhat of the opposite direction. It will certainly help, and there's been some really impressive work on that by Baker and other labs in the past, but I think that it won't get you all the way there, probably.
There are a couple of questions from the online audience. So Alyssa Hummer says, there have been some studies using AlphaFold to predict the structure of a single protein sequence with no multiple sequence alignment, for example, de novo or design of sequences. And could you comment on the applicability of AlphaFold 2? And I guess there are a couple of related questions, which is the inverse problem of protein design, where you know the structure you want, and can you predict the sequence that will get you there? And related to that is another question by Laura Brandt, who asks, could you use AlphaFold to design efficient and specific inhibitors of activators? So there's sort of a set of related questions.
Sure. I mean, I first want to give maybe people a way to think about when will AlphaFold give a good answer. And I mean, I'll first say, if you want to know if AlphaFold gave a good answer, you should look at the PLDDT and the PAE, that the self-reported confidence measures are better than things like MSA depth or any of the kind of things that are correlated with results. But AlphaFold tends to know whether it's given a good answer. But the way that I think about it and the way that we think about it is there are three inputs to AlphaFold, right? There's the primary sequence, there's the multi-sequence alignment, and there's the set of templates provided. And AlphaFold fuses information from all three of those. And so for many proteins, the multiple sequence alignment is strong. It's a deep alignment. It takes only really about 30 alignments for AlphaFold to give a high confidence answer. And so if that's strong, then AlphaFold will give a good answer. But for some proteins, they'll have very, very shallow multi-sequence alignments, maybe only 1 or 2 sequences. But there will be a known template, maybe even a kind of weak template at the 40, 50% identity level. AlphaFold will still give a great answer because that's strong enough for it to use that information together with what it understands about proteins in order to build the structure. And for design proteins, and for proteins that have a very small or non-existent multi-sequence alignment and no templates available, then AlphaFold, if it's a natural protein, will mostly give a poor answer, right? It's a rare case, but certainly in those cases, it tends to give a poor answer. But if it's a design, it gives a very good answer for a simple reason: the sequence is strong. So AlphaFold is a system with 3 inputs, and if any of them are strongly encoding the structure, AlphaFold will tend to find it and will use that piece of information. And so that's how we tend to think about it. And in fact, some of the interesting cases that came up in Demis's talk of, well, AlphaFold ignores templates, and often that's good, is that we have found kind of a priority that if the multi-sequence alignment is strong, then AlphaFold will ignore the templates, even if they're 90+% templates. And in some cases, people have observed actually allows them to correct errors in previously solved structures doing that. For the other questions, I mean, in part, I've kind of answered the design question. For the design of specific inhibitors, I think you'll be in the same sort of situation as designing for structure. Now you're designing for binding. Well, we have a binding system. You'll still be, I think, in this partial case. I don't think it will. I think it will be a very useful tool in the work, but it won't certainly end the problem on its own.
There's a related question to that in the chat box, which is, have you found places where the structures in the PDB look wrong to AlphaFold? You know, how many of those are there? We heard after Demis's talk about errors made by AlphaFold. So this is the sort of reverse question.
So we've certainly, I mean, so when we were developing AlphaFold, the PDB was flawless. And we kind of said all errors are AlphaFolds. Mostly, and we always heard from experimentalists, well, you can't trust everything in the PDB. And we said, sure, but you can trust it a lot more than our half-built neural network. Although really interestingly, and here I'll mostly defer to the CASP assessors who have put out a series of papers recently, and they do this after every CASP, but talking about the different models and what they mean and comparing to the experimental structures. And they have found some really interesting cases where the deviations were in AlphaFold's favor. I mean, one interesting case is, and certainly AlphaFold still makes plenty of mistakes, but AlphaFold's deviations from crystal structures are maybe, I think, in their particular set of 7 proteins, more than half were at near crystal contacts, which suggests maybe the solution structure is different. In at least one case, it was used to reanalyze an NMR structure that had been deposited and a new structure was deposited. So we are seeing some of those, and I think it brings a really interesting machine learning question of how do we now start to jointly think about our uncertainty in both of these methods. And we see some of this in the commentaries from the integrative modeling community of all these methods ultimately have to be fused into larger biological understanding, and it will require each of us to understand more about those kinds of mistakes. And another thing that we observed in our multimer work is that sometimes AlphaFold will produce the structure of not the correct bioassembly in the PDB structure, but will sometimes produce a crystal contact. And, in some of the cases we've looked at, that's clearly wrong. But one of our hypotheses is maybe in some cases, surely the interfaces are assigned incorrectly in the PDB. And so maybe AlphaFold is learning to sometimes produce crystal contacts. And so, and all these things, I think, get to this really, really interesting question where AlphaFold will still make certainly mistakes. Absolutely, and still does make mistakes. But, as computationalists, we have to think more about how to interpret and understand the PDB data. And I think there's really interesting things. And ultimately, now that AlphaFold is a widely used tool, it'll be interesting to see the kind of influence that has on the PDB. And certainly will be part of, I think, before people submit, are probably going to look at the deviations and at least make sure they're supported by the electron or cryodensity. So yeah, it's kind of a weird place to be for me as a computationalist, certainly in structure.
So another question from Jamie Kate is, in the progression through evoformer blocks for the difficult case, there were jumps in the score, and he wants to know what happens in those jumps.
That is a great question. I'm going the wrong direction to answer it. So there is a really interesting thing that, as they observed, so for example, this happens for the most interesting jump, and I'll point, and I apologize to the online audience, but will happen at around block 130. And if we go back to the video of this, and you can see the block counter in the bottom right, and 130 is right about here. So it, I believe, has docked a strand correctly into the structure. And so what you're seeing is over the course of exploration, it has finally found this interface. And then when it has it, it preserves it. And so what we typically see in these traces is jumps exactly like this, but normally monotonicity. Very rarely does it find something and then give it up. And so we see it as kind of a progressive growing when it builds something confident. That's our interpretation. But we think it's individual pieces suddenly being understood when they weren't before. And in this case, might be as simple as a single strand.
Okay, a couple of questions. One is, what are the bottlenecks to RNA structure prediction and possibly RNA protein complexes?
Well, I mean, first we need to try it and find out. Will be first answer. The obvious bottleneck is RNA structure is much rarer in the PDB. And one experiment we really should do and haven't done yet is to say how many structures would we have needed in the PDB to do structure prediction at a certain accuracy. Both is kind of an interesting historical question. When had experimentalists done their job and computationalists hadn't finished? But also from maybe informing how many RNA structures do we need? And it's possible we have enough we need to explore. But I see that as the most obvious bottleneck is simply the number of structures and diversity of structures. I think as we do it, and if we don't succeed at it, then we would expect to see more bottlenecks arise. And my understanding is with cryo-EM, we're likely to get many more RNA structures, which will help quite a bit.
And what were the challenges with the antibody interaction? Considering, the problem of interface is part of the whole problem of complexes, which you've now made some progress with. So.
It's very interesting, and I will first give the answer of ignorance. We don't know. One of the things that it's not, or at least not exactly, is, coevolution is much less important to our multimer system than it is to our single chain, even our single chain system. We've shown things like profile only networks are surprisingly strong with AlphaFold. But for complexes, it's a relatively modest gain that you see by using paired sequences that enable coevolution. So that, to me, mostly excludes that kind of explanation. It probably has something to do, though, with the peculiarities of how antibodies are made, that it must be some different type of interface, or maybe it's a different process, right? They haven't evolved together, and we don't know yet. It could be interface area, it could be shape complementarity. I think it's a really interesting question. I think when we know more about that, it's definitely on our list to analyze. When we know more about that, then we will probably know more about the multimer prediction problem.
Okay. Well, it's 5 o'clock, so thank you very much for a fascinating talk, and thank both of the speakers for really a great afternoon here.
Thank you so much.